Hello. We will start without any further delay to keep on time. It's my pleasure to welcome you to this last session of the morning. This is a session that's dedicated to gene therapy because it's a really absolutely dynamic field in biomedical innovation. And we've chosen 3 topics, all dealing with AAV gene therapy, to illustrate basically an increasingly complex degree of economic valorization and complexity to the path of bringing drugs and treatment to patients. So it's my pleasure to start with the first speaker, who's Dr. Giuseppe Ronzitti from the Integrare Research Unit of Genethon. And he will be talking about AAV gene therapy for Crigler-Najjar syndrome from the bench to the bedside. Thank you.
Bonjour à tous. No, I'm sorry, this was in English, right? It was supposed to be in English. So I'm Giuseppe Ronzitti, as Anne said. I'm gonna give you, before to start, just give you a little bit of introduction about my history and how I came up doing what I'm doing right now. So basically, there is no pointer, okay. But anyway, on the left side, you see that how I started my career. Basically, I have a PhD in biochemistry, and then I moved to a very nice city in Italy, in Genova, to do a postdoc in molecular neuroscience. So basically, I was working on Parkinson's disease, and you know how complex is Parkinson's disease, multifactorial, and so on and so. But we were working on a genetic form of Parkinson's disease, and by chance, I ended up I was put in communication with the world of Genethon in Italy, and in particular, in one of those conventions organized by, no way to get a pointer working there, organized by the Genethon, I realized how powerful could be working in monogenic diseases and how powerful could be the use of gene therapy. And this was, the message was delivered basically by a presentation on the MLD, on the development of a gene transfer strategy on MLD, metachromatic leukodystrophy. So by chance, I decided, at that point, I decided I really wanted to work on this simplified model. It's single gene, you replace the gene, you get correction. That was my very naive actually, hypothesis at the time. By chance, Federico Mingoaz, who is here in the picture, I'm sorry, I cannot point out, but it's here, you know, where there is the arrow. He was coming back from the US, and I decided to join his lab, and I was lucky enough to be able to join his lab. So I moved in France, 2013, and I started working basically in gene therapy for metabolic and rare diseases. So the lab was basically not existing at the time. It was founded in 2012. There was nothing there. When I arrived, there were we were 3 people. And I can just, the foundation was in 2013. Up to 2017, Federico was with us. Then I get the team, and I continued all the story and all the experience and the scientific activity in the lab. But there were 2 pillars basically that were actually sustaining the activity of the lab, and these 2 pillars are not really very much innovative. It's about science and money. So excellence in science, this is important. So the good papers made out, good article, good science made in the lab. And on the other side, the access to a competitive grant. I think there is nothing more to do to have a lab working in a very efficient condition. So you see, what is important is also to be constant, to get this access to funding and to publish good papers for the whole period. So as I said, the lab is focused on the metabolic diseases, and of course, metabolic disease exists for any type of organs, but we focus in particular on the liver. And we work on the liver for one simple reason, which is the delivery advantage that you have in the liver. So this is dependent on the structure of the liver. Basically, what you have, whenever you have, okay, the liver is very much irrorated by blood, but what is really makes a difference compared to other organs is the endothelium, which in the liver, once again I cannot point, but in the liver it's discontinuous. So you have basically gaps into the endothelium. So whenever you have a molecule, whenever you have a vector, the vector can enter the liver, can really get in contact to the hepatocytes and so transduce them, and so you may have a definite advantage. So, but the liver is not just about of course, you're not working on a tissue delivery just because of you have an advantage. You work because you have a real and clear clinical need. And in fact, you do have this. You have multiple diseases associated to the liver. And one which was particularly associated to hepatocyte dysfunction is this one, which is called Crigler-Najjar syndrome. So this is a ultra rare, really ultra rare disease. One person in 1 million at birth. So you imagine there are like few cases in France, one out of the 2 years, basically, born with this disease. It's a real and clear deficiency of a liver enzyme. So it's an hepatocyte enzyme specifically expressed there. Whenever you miss this enzyme, what happens is that you cannot process bilirubin. So bilirubin is coming from heme. So it accumulates, and when it accumulates, it can get toxic. There is also another characteristic of bilirubin that makes it interesting, is that it is yellow. So that's, you can see the people. I don't know if you can appreciate from here, but the people with this disease, they become yellows because bilirubin accumulates into the skin. So, I mean, the color is not, of course, the color has never been an issue, but the thing is, whenever you have accumulation of bilirubin into the tissues, what happens is that it can cross the blood-brain barrier and then end up in the brain. And it has a huge neurotoxicity. So you can end up with these patients. If the levels of bilirubin are not controlled, you may end up with patients dying or having important neurological symptoms. So there is a cure. I think you know that. I mean, you have seen probably you have seen that happening in the clinic, because jaundice, which is the manifestation of the accumulation of bilirubin, is something that is happening typically in neonates. So the treatment in this case is phototherapy. So basically you put the babies under the lamp. The lamp will convert bilirubin in a photoisomers, and the photoisomers can be excreted by urines. So this is, of course, for a neonate is quite easy as a treatment, but can you imagine trying to get an adolescent under a lamp for 10 to 14 hours per day? So this is very, of course, the compliance is not as good as possible, so the bilirubin level get high, and so you may have toxicity in the patients. Of course, the phototherapy works, but it's a symptomatic treatment, so you are not basically going into the cause of the disease. There is also another issue with phototherapy, is of course that the surface to volume ratio, of course, decrease with age. So the phototherapy basically gets less efficient. And then the only treatment, as I said, this is a real hepatocyte disease. So if you transplant liver, that's a real treatment. So the patients with a transplanted liver becomes totally normal. They start degrading bilirubin, and then they stay normal. So, but of course you will have another disease, because you will be submitted to immunosuppression for the whole life. So based on all this consideration at the time, this was 2012, 2013, once again, we decided to go for a gene therapy for Crigler-Najjar. So an AAV gene therapy. At the time, the AAV8 was one of the best performers for liver gene transfer. And I still remember, I mean, the call we had with Federico. He told me, okay, you come here at Genethon, we can start doing Crigler-Najjar, and then we go ahead with that. With other diseases, we will see. There was Pompe disease already. There was something planned already. But anyway, the idea behind doing gene therapy with AAV8 is quite simple. Basically, you end up with expressing the enzyme which is mutated. In this case, it's UGT1A1. It's, the name is quite complex, but you express this in hepatocytes. Then the activity is restored, so you can start eliminating bilirubin. So the levels of bilirubin will go down and will stay controlled over time. So that's the rationale, but it was not easy as as we imagined at the time. As I said, what is interesting also to know is that the proof of concept study, so it was already demonstrated in 2006, it was already demonstrated that a treatment could be developed. So there were rats treated with a vector, an AAV vector, and they were correcting their bilirubin level. But it took time, I mean, to go to the clinic, and you see that we get the project back in 2012, 2013. So we work with very important, we had a very collaborators that had developed an alternative model of Crigler-Najjar, so a mouse model. We had the rat model internally. We decided the candidates in 2014. We went to the large scale process development, which is something that takes much time also. And then we did all the studies. They were what I called the IND enabling studies. And then we started the clinical trial in 2018. So from the proof of concept to the clinical trials, it's a 12 years time. And this was an easy disease. This was an easy disease. So you can imagine the complexity of doing that. Of course, we are getting better with time. We can shorten much of the time, but the thing is, it takes time, and it takes money. That's the other issue. It takes a lot of money, which are coming, of course, from the AFM Teleton and Genethon, that you know that, but also from the European Union. So there is a grant which is called CureCN, which gave us, a good amount of money to do some research also on the same context, but also to really implement the clinical trial. So that's really important. And it's also important to start with models, because this is very important. I will come back to this. I'm showing these results. This is Fanny, basically. It's very important because she started with me, basically, the project. We started together. The project in a single room. We had a single room in the lab, and we started this project. And so these data are the data of the dose finding, which in most of the cases, they are totally uninteresting for a researcher. But in fact, this year, I want to show you the dose finding data in Crigler-Najjar rats, because you see that what happened, well, I cannot point, but you see that the lowest dose is the one that you have like a broken line. You see that there is a decrease in the bilirubin level in these rats. So these rats recapitulated the human disease, a decrease in the bilirubin level, then it goes up again. So indicating that there is something happening there that may be important for the dose finding then in humans. And I will be back to these results afterwards. So we planned for the clinical trial. This was quite long, but in the end we ended up with this clinical protocol where we had two doses, basically two quantity of vector administered. It's 2 times 10 to the 12 and 5 times 10 to the 12. I mean, the dose doesn't matter. It's not a high dose. It's a dose that is quite safe to be used in humans. And we had the primary endpoints. This is a phase 1/2 clinical trial. Basically, the primary endpoints was clear. Okay, there was safety, of course, but there was the efficacy inside. So the safe interruption of phototherapy at week 16. So the goal was really to stop the lamps, basically. That was the idea. And so that's what happened in the first dose cohort. So we had planned 2 patients, 3 patients actually at the very beginning. But you see what happened is the left part of the curve is quite important, because you see the levels of bilirubin in patients with phototherapy. So they are quite high levels. Consider that in a classical individuals with no jaundice, you have level of bilirubin at 0, basically. You don't have any level of bilirubin. And then you see that these are quite high levels, but they are still controlled with phototherapy. After the infusion of the AAV, what happened is that you see a drop in the levels of bilirubin. And then, this was quite, I mean, it was not really satisfactory, but what happened is that the levels, they stay down for a certain period, and then they start to go up again. So this was exactly what we have seen in the rat model. And this was very important, because based on the results on the rat model, where we haven't seen any immune response, we thought, okay, this is just the model. It's just the disease where you have this kind of feature. So it's not an issue of the treatment. The treatment works, but the dose is not high enough. So we were able to go up with the dose, and this is what happened. So once again, the level of bilirubin are quite high before the treatment. After the treatment, we see a huge drop in the levels of bilirubin, which goes down, stay down up to 16 weeks. Then the clinician, based on the level of bilirubin that we have here at 16 weeks after injection, they decided that it was possible to stop the phototherapy. They stopped phototherapy, and you see that the levels of bilirubin, they stay down for at least 24 weeks in this graph. So we, of course, we have a follow-up on this study. Well, yeah, that's something that I always forget, but this is also something nice, which is about the way the patients are at this point when they have low bilirubin. So basically, they look normal, which is not something trivial for patients. And it's also allow me to discuss also another thing, which is the importance and the relevance of having endpoints. Yes, I'm getting there, and you know so the relevance of endpoints. So having good endpoints to evaluate the efficacy of a treatment. This is, I think there is no another endpoint like this one in any other disease where you can really see the results by looking in the eyes of the patients. And so there was, of course, there was the first time an activity, an enzymatic activity in the liver was restored with an AAV8 vector. So we were able to publish in New England. This novelty was now published in New England most recently, basically this summer. And of course, the conclusion, these are the conclusions related to the clinical trial, which are anyway important because also there is we have a follow-up in the paper, in the New England paper, up to 8 weeks. But another important thing is to keep good relations. I think this is the most important thing, is keep good relations with anyone in the field, and having the possibility to discuss all the results with good friends and collaborators. And speaking of collaborators, somebody once said that it takes a village to get to a clinical trial. That's the village, that's the map of the village. You see how many people, well, I cannot tell how many people in the Genethon R&D are working on this program, but there are also collaborators, external collaborators, starting from the scientific part, the central one, and then the clinician, the patient association, the production, which is not trivial once again. So it's, in both cases, he was producing for us the thing, and of course, monies that are always there. And then I finish with also the acknowledgements, the internal acknowledgements of the team, of course, all the people on the team that worked, and of course, all the agencies that support our work, and all of you for your attention. Thank you.
So it's a mystery. When it's green, they don't work. When it's red, they do. I don't know.
Strange.
Yeah, I don't know how they work.
It's green, so it doesn't work. Okay, thank you, Giuseppe. Very, very nice. So one is just a remark, I guess, because it's a rare disease, right? I guess for you enrolling many patients is a problem, and in fact, that's why the numbers were low. And I don't know whether, you know, this impacts your going farther, because maybe you need to enroll way more to finally get statistics about it. But this is just a remark. The second thing is, did you try to also use only the AAV treatment without the phototherapy at all? Because it looks like you have it, then you stop it, and you see it stays down. But did you try to use it as a single avenue, if you want? So to know whether it's a combined, is a synergistic effect of both, and then it stays down, or is it because of your treatment? And the second, the second question is, are you thinking of doing protein engineering to make the protein more potent?
So I will take the last question. Protein engineering, of course, is the best way to decrease the dose of vector, and so decrease the cost, and then go ahead. We are doing some work on other diseases where we are doing. Of course, the protein engineering has also a drawbacks, which is the immunity, because you are changing the protein, so you may end up with either an unknown activity of the protein in humans, I mean, or a immune response to the protein because it's different. So it's always about that. But we are doing, for instance, in GSD3, we are doing that. We are doing protein engineering to reduce the size and fit in a single AAV. So that makes sense. The first one was about the number of patients. That's clear. It's any rare disease. There is nothing to do. It's very hard to get patients. There are criteria of inclusion, so this will reduce the population. So this, of course, is one of the main and biggest challenges in the development of a gene therapy for a rare disease. And the second one, without phototherapy, so that was a safety thing. So basically, you cannot convince a clinician that you can switch off phototherapy and give gene therapy. That will not gonna help a lot. But it's true that in animals we do like that. In animals where the toxicity is less of a concern, rats typically don't die for increased bilirubin. So there we do that without phototherapy and it works pretty well. The synergy exists because the pathway actually that degrades bilirubin are totally different, basically. So the synergy, for sure, it exists. What is interesting is that in the New England paper, we demonstrated that the synergy will not occur immediately. Basically, it takes time for the bilirubin to go up a little bit because of the absence of phototherapy. It's not just immediately that it happens. So it's probably related to accumulation in the tissues and then repartition into the blood. So it's a little bit more complex. These are things that, of course, they were not modeled at all in such a rare disease. So, but yeah.
Hi, thank you for the very inspiring presentation. I was wondering, since it seems to be a both pediatric and adult disease, how did that impact like the progression through clinical phases and things like that?
Well, that's quite clear, and the phase 3 basically will include and is including pediatric patients. That's of course you have to do that. But there is another drawback, which is you cannot dose AAV at any age, because basically whenever you, in particular in the liver, if you dose AAV at very pediatric, like 0 to 2 years, the risk is that you will lose the efficacy. So basically we convinced the agency that this was not a good idea to go for a pediatric phase of the trial with 0 to 2, and we still have to convince them that we will go up to 10. But the idea is to dose patients at 10, so the liver is developed enough to don't have any dilution. But of course, yes, it's a pediatric indication, as you said, so we have to plan something on the... It's not approved yet. It's just a clinical trial, that one, yeah. But it is that we started with, you should start every time. If the disease is not life-threatening, you start with adults and then you go down to pediatric. Yeah.
One last question.
Thank you very much for the nice presentation. I was wondering, because here you use the adenovirus, and you know that after a while you will lose the expression of in this case, of your gene of expression. So did you think about using lentivirus for do a permanent insertion and avoid the constant? I know that it probably will be much more expensive in the beginning, but lentivirus is already approved for gene therapy. So what do you think about it?
I mean, that's a good question. These are adeno-associated viruses. That's the first thing. I'm sorry, I have to correct you, because the adenovirus is not a very good story. But adeno-associated have shown some persistence, so that's one thing which is interesting. They show persistence, at least in hemophilia, for up to 10 years, I would say, in a tissue which is slowly replicating. Of course, your question is very nice, because whenever you have a condition, a liver condition, where you may end up with higher proliferation, you may lose the efficacy very, very fast. So the idea to use lentivirus is very much, I mean, there are companies developing that for liver diseases, but I think they will be tested at first in liver diseases where you do have these components of high proliferation, so the AAV will really not work at all. So there is a scale, basically. You start with the most severe, you see what happened, and then you go down to the less severe. Maybe yeah.
Thank you very much, Giuseppe.